tract in just a moment and actually look at the way that that tract enters the cord and carries information up to the brain. By contrast, motor tracts, as we've seen, end with the word spinal, so corticospinal, rubospinal, thalamospinal, and so on. We also mentioned that while there are gray matter tracts, we're primarily concerned with white matter tracts, and that with the exception of these tracts, which we're also going to look at today in more detail, the dorsal column tracts, it's sometimes very hard to predict on the basis of where the tract is located, whether it is sensory or motor. So don't assume that there's a simple relationship. In general, today, we're going to be focusing on these two, at least at the outset, focusing on these two critical sensory tracts. One, probably the lesser of the two, the dorsal column medial lemniscus system, dorsal column, and that terminology indicates where the tract is located. And the other tract I mentioned is the spinothalamic, but it's also called the anterolateral system. Anterolateral. So here's your spinal cord. Is ventral or anterior at the top or the bottom? So this will be down here, and lateral is that way. So your anterolateral tracts are going to be sort of in this area. Not exactly where you'd expect them to be if we're talking about a sensory portion of the spinal cord. So again, I would expect that you'd be able to identify at a cross-section of the cord the dorsal columns. We've looked at these several times. And the anterolateral. And you'll see different representations of the spinothalamic tract because it will have a different appearance depending upon the transection that you're looking at. But at least you understand how the term is derived. So these are the two tracts we're going to be focusing on, the dorsal column and anterolateral. And I've listed here some of the important differences between the two. I think we've made this distinction before, that it's both an anatomical distinction and a functional distinction. So if you look at the dorsal column system, notice the sensations that are conveyed, in particular, proprioception. And we said, we've made the point on several occasions, that proprioceptions take preference, they take priority over pain and temperature sensations. And when I say they take priority, what do I mean by that? What does that mean in reality? Yeah, they're faster, they utilize larger fibers and so on. And you can see that in the types of fibers that we use. So I've also described the anatomical differences, but I think it would be better just to look at them here. And there are many subtle variations of this we won't need to worry about. But what I want you to pay a look at the medial lemniscus tract first. So here's going to be the sensory neuron. Here's the free nerve ending of a sensory neuron. You know the morphology of this neuron. It's a somatic sense, therefore it's a tactile sense, it's touch. Therefore, what is the morphology of this neuron? What's the shape? Yeah, it's a unipolar neuron, and the cell body is going to be located in a DRG, relatively proximal. Remember, the cord is much shorter than the column, but relatively proximal to the site of the stimulus. So, by the way, all of our terms, when we talk about how the information is ascending to the brain, it's always with respect to the stimulus, always with respect to where the sensation, probably should just use the word stimulus, is initiated. All right, so let's follow this track. Here's our sensory neuron. Here's the axon going up. Now we're in that dorsal column. Remember, this system is also called the dorsal column medial lemniscus tract. So that's where we are. We're in the dorsal column right there. you see any synapses? I don't see any synapse yet, so we're going up, going up. It ascends ipsilaterally. And notice where we are now. We're already in the brainstem, we're already in the medulla. So this neuron has axons that extend all the way from the skin to the brainstem, one neuron. And notice also in this system the ascent of information. The action potential ascends ipsilaterally. We used that word before. Do you remember what that means?
Same side.
Yeah, on the same side. Ipsilateral. So we say the DCML ascends ipsilaterally to the medulla. Its synapse is there. I'm a little hesitant to use the traditional terminology. This is sometimes called a first-order neuron. Then its synapse is with a second-order neuron in the medulla. And what happens to the second neuron? What does it do? Right. Well, what is this? What's happening right here, Brian, through the line? Yeah, crosses over decussation. So there is immediate decussation in the medulla. And you're right, we're in the lemniscus now. And then from that point, it ascends contralaterally to the thalamus. So the secondary neuron in the DCML, the dorsal column medial lemniscus tract, the second neuron decussates and then ascends to the thalamus. And then the final neuron, which in this case we'll call a tertiary neuron. Again, I don't really like this terminology because it's not used consistently. Simply extends from the thalamus to the sensory cortex. And that will be true for the other pathway as well. All right, let's look at the spinothalamic tract, or again has the other name, anterolateral system, and see how that compares. Once again, we have a stimulus. Here's our unipolar neuron. Stimulus is on the same side that it was in our illustration of the DCML. Here's the cell body of the afferent neuron, still a unipolar neuron. But what's the first big difference you notice? All right, that sensory neuron is not very long. That initial neuron immediately synapses in the dorsal horn of the cord. This is why I don't like this terminology, because again, it's not used consistently. So we'll just call this the first neuron, or the primary neuron. Primary in this sense meaning the neuron that's actually responsible for the sensation. Recall in both of these, well, probably not the best example because this is going to be pain, but in this case, the neuron is also the receptor. This is a free nerve ending, and it's going to be responsible for this. I think is a Bunsen burner or something. The person puts his foot on a Bunsen burner. So that's a flame that you see there. All right. So in the dorsal column, there is a ipsilateral synapse, and then what does the next neuron do immediately? Decussates, so there is immediate decussation. This is not quite anatomically true, but for the purpose of our discussion, we'll use the following terminology. We'll say that there is decussation at the level of the stimulus. Remember, the cord is shorter than the column. And certainly your cord, neither your cord nor your column extend all the way into your foot, but you understand what I'm saying. At the same nominal level. All right. And then that second neuron carries the signal, and you tell me, where does it terminate? Yeah, in the thalamus. There's no synapse in the medulla.
Does the second neuron, when it decussates, does it stay inside
Well, this is not really gray matter. This is actually a white matter tract. They're showing that it's going out to that anterolateral system. That's actually a white matter tract. So does the spinothalamic tract ascend ipsilaterally or contralaterally from the stimulus? All right. It ascends contralaterally in the cord all the way to the thalamus. And then there you have the same arrangement that you have in the DCML, a neuron extending from the thalamus to the primary sensory cortex. Yeah.
So by saying it the same way, there's a synapse in the thalamus and then from the thalamus, there's a tertiary neuron.
Yeah. Excuse me. We have to be careful with our terminology. I'm using the adjective tertiary because it doesn't really mean anything except it's the third neuron. The traditional terminology, you can see it here. Axons are first order neurons, not part of the spinothalamic tract. This is archaic and it's just going to confuse you. So I would encourage you just to think about this as being primary, secondary, tertiary, and leave it at that. I'm not really interested in the arcana of anatomical classification. Yeah.
The medial lemniscal tract, is that the tract from the medulla to the thalamus?
Well, certainly the medial lemniscus is here.
Oh, in the medulla.
Right. So we call the entire thing the DCML just because it involves the dorsal column of the medial lemniscus. In the same way, anatomically, that we describe this as anterolateral. It's simply an anatomical clue.
Is there a name of the place in the medulla where the first and second neuron synapse that we need to worry about?
No, and it's going to vary. It's going to vary with the nature of the sensation and the receptor or the pathway. So that's not terribly important. But what is important is what we talked about back here. Make sure you know the critical differences both anatomically and functionally. And I have another little table of the functional differences coming up in just a moment. And this is an illustration showing the same thing, the DCML on the left, spinothalamic on the right. In fact, on the test, I might pull one of these from an entirely different source, but based upon your understanding of the anatomy, you should have no trouble differentiating these two, based upon your understanding of which one ascends ipsilaterally, which one ascends contralaterally. So we've pointed out the DCML system conducts more rapidly, has larger fibers, tends to be more myelinated. We've indicated that it ascends ipsilaterally, whereas the spinothalamic tract ascends contralaterally. And then in terms of function, again, just a very, very gross sort of simplification. Position sense, very, very fine sense of touch. In other words, we were doing two-point discrimination, and that's a little more complicated than this because it involves specialized receptors we'll talk about in just a minute. But for very fine two-point discrimination that you would use, say, in your fingertips, that would be DCML. Again, whereas the spinothalamic tract, very crude touch, mostly pain and temperature, tickle and titch, as I call them. They were famous. How many of you were? But tickle and itch, and then very crude pressure sensation. So clearly, high priority, lower priority, phylogenetically more recent, phylogenetically older. And we've made this point several times already. All sensory pathways convey information to the thalamus with the exception of the olfactory pathway, which goes right to the limbic system. And we could quibble about the equilibrium pathway. In fact, we could quibble about the, Luis made the point about the olfactory pathway. It's important to understand that by secondary pathways, all sensory information ends up in the thalamus. By secondary pathways, olfactory and gustatory information also end up in the thalamus. It simply means that the direct projection of the olfactory nerve goes back to that entorhinal cortex and the perirhinal cortex and so on. There's nothing new here. You remember our discussion of somatotopic organization? What was the definition of somatotopic organization? Yeah, Trenton was pointing out that by somatotopic organization, we mean that the entire nervous system, motor and sensory to varying degrees, the entire organization of the nervous system is, at least to some degree, predicated upon, in many respects, similar to the organization of the body. And we saw that in the somatosensory homunculus. We can see that there's an additional body part we did not see before that clues us off to the fact that this is a sensory homunculus. But because I'm a gentleman, I'm not going to point that out. Although it is in an odd position, right? And it's, would you? Just saying. So that's what we mean. And then, of course, remember that sensory integration is just the opposite of the primary motor integration. Remember, by that, in the primary motor cortex, that begins the final common pathway. That's the upper motor neuron that sends the information to the lower motor neuron that causes the muscle to contract. But these pathways we've been talking about, the ECML and the anterolateral, where do those pathways initially terminate? Where does that tertiary neuron terminate in the cortex? Primary, supplemental, yeah, primary sensory cortex. So it's just the opposite. On the sensory side, behind the central sulcus, the sensory processing begins at the primary cortex. It doesn't end there, it begins there, and then diffuses, again, in a very simplified fashion, posteriorly. Yeah, there's so many complications to this that I just don't think it would be terribly productive to discuss all of these right now. You probably saw, I should have brought some water. You probably saw the recent experiment or the recent study involving the man who was quadriplegic, but he was able to, with a little robotic arm, bring a drink to his mouth using implanted electrodes in his brain. They took a little disc and just put a little disc in there. And with that information, which is again a crude source of information, was able to, this is the future, this is your future, was able to take the robotic arm, and by thinking about what he wanted to do, and the way they did this is incredible, he was able to take a drink the first time in his life by an act of will on his own. But what brought that to mind? Have any of you heard of mirror neurons? What are mirror?
They're, I've heard described as like sympathetic neurons. You can, your brain fires neurons that allow you to feel what someone else feels.
Yeah, good way of putting it, actually. Yeah. When they were discovered, and they were actually discovered in monkeys, when they noticed that the monkey with the little electrophysiological electrodes implanted in the brain were watching one of their caretakers eat some ice cream. And they noticed that the areas of the brain being discharged in the monkey brain corresponded to the same motor areas that the caretaker, part of the human brain that's responsible for the motor actions of eating an ice cream cone. And how would you explain that? Well, as was suggested, there must be some sort of commiseration or sympathy. Depending how far you want to take this could be the basis. This could be the cerebral basis for compassion and other aspects of human endeavor. One theory of autism, for example, is a dysfunction of mirror neurons. But in any case, the reason I was getting into that is that although these are called mirror neurons and involve mostly motor pathways, a lot of these mirror neurons are in what we call the sensory brain. So when they were working with this fellow and trying to get this robotic arm to follow his commands, his will, a lot of the electrodes, a lot of the information was coming from the sensory portion of the brain. So it's really a little more complicated than our explanation involved. There's been a lot of recent pushback against the whole notion of mirror neurons. I think neuroscientists are getting a little fed up with everything being attributed to mirror neurons, and we don't read about them as much as you did a few years ago. The only purpose of this slide is I want to remind you that in the face, you have a similar sort of sensory apparatus. We've been talking about sensations arising from the body. But you know that, for example, sensations arising in the face, anterior cranium, generally involve what cranial nerve? 5, yeah, 5. And so notice, we have a good picture here. Look right there. Can anybody, because you don't have to, I'm not going to ask you for a moment to make sense of this very complicated diagram. But what have I circled there? Is that the DRG, right? The DRG. The trigeminal, see, that's the trigeminal ganglion, semilunar ganglion, gasterian ganglion. And what do you notice about these neurons, the morphology of these neurons? What type are they? Unipolar. Unipolar. So that's why I was using that terminology. The trigeminal ganglion, in many respects, is just a sensory ganglion, and then the other side of that axon, we'll call it the afferent side, is then projecting to cranial 5 nuclei in the brainstem. Remember our distinction between ganglia and nuclei, very important when we're talking about the sensations from the face. This is cranial nerve 5 ganglia here. Here's the nucleus here. This one of the nuclei. You don't need to worry about the pathway. I just wanted to show you that there is an analogous organization. One of the differences is, though, that these facial pathways tend to be more bilateral than do the sensory pathways, bringing information from the body. And notice you also, too, have a nuclear differentiation, a prioritization of proprioceptions over pain and temperature sensations as well. But again, that's all I would mention. You don't have to worry about ciphering this complex anatomy. We've talked about a little bit about specific receptors. Remember, at this point, we're talking exclusively about somatic receptors. A mechanoreceptor, for the most part, is just a stretch receptor. It's sensitive to some type of physical deformation or displacement. Thermoreceptors respond to temperature. And then nociceptors respond to pain, again, although we need to talk a little bit more about that. There are a number of different types of sensory structures in your skin, for example. You can see something here called a Merkel's disc. Now, note that a Merkel's disc is actually a discrete receptor. So here's where things get a little more complicated. With free nerve endings, the primary neuron is also the receptor. But in the skin, for some types of sensation, the body uses specialized dedicated receptors and then conveys that information to a succeeding primary neuron. Merkel's disc says here is slow adapting. What does that mean? Somebody break it down. What is that?
descriptions of the action potential.
All right, so explain it in such, somebody explain it in such a way that that I know what that means. For example, what is adaptation?
Well, it senses the initiation or termination of a stimulus.
All right, so is that slow adapting or fast adapting?
That's slow. No, that's fast, isn't it?
That's a high probability answer, but not just. So you said that you gave me a definition where the receptor senses only the initiation and the conclusion of the stimulus. Is that fast adapting or slow adapting?
Fast.
That's fast adapting. Right, good. And your password today was phasic. So that's a phasic receptor. All right. But a slow adapting receptor, this is pretty much saying this: as long as the pressure is on the skin, that whatever your brain does, that neuron is going to continue sending action potentials back to the brain. It's slow adapting. It's slow to adapt to the stimulus. Whereas a fast adapting receptor, which wouldn't be this type of sensation, initiation, then nothing. I remove the stimulus, and then nothing. So it's only, the fast adapter is only responding to the changes in the excitation of stimulus. So Merkel's are slow adapting, but they are specialized receptors. Now what we're looking at here is is a taste bud. And it's not very clear, but this whole thing here, we can't really see it, but that whole thing would be a papilla on your tongue. And so here, this little thing here is one of these little taste buds that lie on the wall of the papilla and respond to, as receptors that respond to various chemicals, molecules, or such. Rufinis also respond to broad deformation of the skin. They're also slow adapting. So we have a picture. Here's a Ruffinian corpuscle here. The free nerve endings, we said, are a little bit different. These can turn up all over the place. They can appear up here in the epidermis. These are about the only type of receptor that extend all the way into the epidermis completely. You can see that the Merkel discs are right there at the interface of the basal layer, but these free nerve endings can go way up into the epidermis. So sometimes you find them there, sometimes you find them wrapped around the hair, the little hair follicle. Yeah.
Are all these these receptors, the Physio and the are they all slow adapting?
No. In fact, the Pacinian is an example of a very fast adapting receptor, and its ability to adapt very rapidly. That's not one there. Here's a Pacinian corpuscle. Its ability to adapt very rapidly enables it to provide you with sensations of vibration. We talked about, for example, why do you have fingerprints? Anybody? The idea, yeah, you run your finger across an edge and like the washboard, and it's the Pacinian corpuscle perhaps that is conveying, or at least assisting in the conduction of that information, generation of that information to the brain. Meissner's, you can see a Meissner's corpuscle here up in the papilla. Meissner's also rapidly adapting to provide you with the 2-point discrimination that we talked about. There'd be a high concentration of Meissner's in your fingertips relative to your back, for example. What is the definition of cold to a physicist? Right? Yeah, the absence of heat. It's not that simple in our nervous system. We actually have specialized receptors for different temperatures. In general, we have pretty largely 2 different thermoreceptors. We sense cold between 10 and 25 degrees Celsius. And we sense warmth between 25 and 45. If it's colder than 10 or hotter than 45, what do we sense? Pain, yeah. There's a lady undergoing a sobriety test. This is an example of proprioception. And again, this type of information is conveyed by a number of little sensory structures, chiefly, when the eyes are closed, we're not using any special sensory information in this type of proprioceptive monitoring, but chiefly by muscle spindles that are sensory capsules embedded in your skeletal muscle, and then Golgi tendon organs, so that when you do the patellar reflex tendon, you can see how robust mine is. That would be located in the tendons and responding to changes in the tension, the stretch of the tendon. So you can see this person is dramatically stretching the hamstrings there. This is the little spindle that would be embedded in the muscle. And so that's going to convey sensory information back to the CNS. This is a little bit simplistic here. And then that might result in, hey, you need to, in order to avoid tearing or stretching or damaging this muscle, it's going to be necessary to relax a bit, so that information is then conveyed back to what are called extrafusal fibers. The contractile fibers of skeletal muscle are called extrafusal, E-X-T-R-A-K. The muscle spindles are called intrafusal, and these are the sensory organs. They go by a variety of names. Yes, the skeletal muscle, you can see it here, the contractile skeletal muscle over here is called extrafusal. The sensory bag, the little spindle organ that you see here, that's called an intrafusal fiber. Remember in the anatomy we talked about the fact that those spindle organs also receive motor signals. Have we talked about that? So these are what are called gamma efferents. But notice this, axons of gamma motor neurons. And what these are doing is that these gamma motor neurons, these are efferents, these are motor signals going to the sensory structure. We're going to see this again and again, or you will, in your continuing study of these matters. These motor signals, again, this whole thing is a sensory structure, but these motor signals are dynamically changing the dimensions of this to maintain its ability to provide accurate sensory information to the brain. If we stretch this thing out completely, as it would be in a stretched muscle, it no longer has the ability to respond adequately. So what would these things do? They would tell it to shrink, so you maintain your sensitivity to muscle tone. And of course, this is going on. There's this exchange of information dynamically between the afferent and the efferent fibers, gamma motor neurons that are present in a muscle spindle. Remember, I used the example of having a level. Probably best you don't remember that, but the level is going to, as I move my arm here, I'm going to level my arm here. Now, if I move my arm, the level's no longer going to be in the center, so I have to move the level. Well, that's what these are doing. They're changing the position of the level each time so that it reads properly, so that it provides accurate information.
When it says it enhances the sensitivity of the receptor as muscle length changes, is that a direct relationship? Like it becomes more sensitive as it becomes longer or shorter? Or not directly?
Excuse me. I think what is implied there is that the gamma motor efferents are constantly changing the length of the muscle spindle, again, not so much to enhance or decrease, but to maintain the constant sensitivity, regardless of the tonus or the stretch of the muscle. So we talked about nociception. We indicated that in many respects these are pain receptors. In fact, we've already talked about where pain receptors are located. For example, in the nervous system, we indicated there. You don't have nociceptors in the parenchyma of the brain and the neuropile, but you do have nociceptors in your meninges, especially in the dura. And that's, as a PA, that's going to be very important. From now on, you know, one of the primary complaints of your patients will be about pain, and you therefore need to know the physiological substrate of that pain, where it can be, where it cannot be, and all the complexities therein. There have been several studies since this, but there is some evidence to suggest that swearing relieves pain. I know it relieves the anxiety of test taking too, because I hear those sounds in the computer room. But there is a difference between pain and nociception, because I'm usually not doing so well talking today. Nociception implies a physiological substrate, a physiological basis for the pain. But remember, we talked earlier about the fact that not all types of pain have a physiological substrate. What types of pain would not have a physiological substrate? Well, that may or may not. That just means we don't know. But it could be. That could be the case. Yes, I think you're right. Psychological pain, I think that's very important. And we talked about phantom pain, right? What is phantom pain?
Non-existent.
Right. The pain that accompanies loss of a limb can be absolutely debilitating. So again, that's the primary distinction between pain and nociception. Pain is a little more complicated. But nociception does require the presence of nociceptors. There has to be nociceptors. These are usually free nerve endings, as we've talked about, that are conveying these afferent signals to the CNS. Any number of things can be associated with pain. I've listed some here. Inflammation is generally painful. There's a whole sequence of events involving a variety of chemicals you're going to learn about later. Ischemia is often accompanied by pain. What is ischemia? Yeah, impaired blood flow, reduced blood flow, or abnormally reduced blood flow to a tissue. Muscle spasms, a lot of the back pains that you'll be, I think I read not long ago, that a third of the complaints in the emergency room involve lumbar pain. Well, oftentimes that muscle spasm is secondary to some other problem, but it's the spasm that's actually the cause of the pain. Nociceptors have been classified in a variety of years, in a variety of ways. In my lifetime, I bet they've reclassified nociceptors in at least 5 different ways. So we're just going to do this very conservatively. We'll talk about thermomechanical pain, which is often considered fast pain, and then slow pain, which is sometimes called polymodal and can be caused by a variety of stimuli. Fast pain is faster. It's coming to the CNS by means of A delta fibers. The slow pain is ascending primarily by unmyelinated C fibers. And if you go back to that little table that I ripped off from the Wikipedia there that you have in your slides, there's not much difference between these two. They're both pretty slow. Remember that your nervous system prioritizes proprioception over pain anyway. Of course, what is an analgesic? Yeah, analgesic is just a pain reliever. What is hyperalgesia? Yeah, an intensification of the pain, and both are going to be important to you. But indicate here that nociceptors are sensitized by the presence of prostaglandins. Prostaglandins are lipidic, immunological, I don't know what immunological, they're lipidic hormones, except they're not even really hormones. We haven't talked about the way that, how can I say this? These are chemicals released locally that act locally for the most part They really don't live long enough to last in the bloodstream. They get in the bloodstream, but they don't last very long, so they're really not like hormones. And there are at least 10 different prostaglandin receptors. They do a variety of different things. But one thing they are involved in is the sensation of pain, or at least they intensify. They can intensify, especially inflammatory pain. And there are a lot of analgesics. You see that NSAIDs here. What are NSAIDs? Yeah, non-steroidal anti-inflammatory. These can sometimes act to inhibit the enzymes, the cyclooxygenase called COX, that produce the prostaglandins. And so that's how the NSAIDs work, by simply lowering the levels of prostaglandins, these pain-intensifying hormone-like substances. As I say, I'm not too concerned about the terminology we use to describe pain. Fast pain, again, described as sharp, pricking, acute. Slow pain, burning, aching, or throbbing. But recall, these involve anatomically different pathways. Fast pain, A delta, slow pain, C fibers.
What does it mean by upper tissues?
Where do you see this?
Fast pain generally restricted
Number
to
3.
upper tissues.
Yeah, I see what you're looking at, and I have absolutely no idea. I think it may be because I'm burdened by my debility at the moment, but I'll think about it, see if I can figure out what I was talking about there. In any case, I'll get back to you on that. Nociceptors tend to adapt slowly. We've talked about what they do. So, and this is a problem because I don't know if you've ever had, imagine a thorn, a cactus thorn in your foot or your hand. You remove the thorn, but the pain persists. Pain perceptions, nociceptions often persist long after the stimulus has been removed, and that persistence has a neurological basis. Now, we've talked about the fast and the slow pain. The fast pain is generally along a pathway called the neospinothalamic, the slow pain on the paleospinothalamic. And once again, just very simply note the following. Note that the fast pain pathway is predominantly glutamatergic. Fast pain pathway is predominantly glutamatergic. Whereas the paleo pathway, the slow pain pathway, the older pathway, involves both glutamate and substance P. Substance P is a proteinaceous neurotransmitter. And as we pointed out, the slow pain is more diffuse, it's duller and much less well localized. The body has a variety of endogenous analgesics. I don't know if I have that word there or not. I ran out of room there. And then I ran out of the ability to. Oh, thank you. Thank you. Where do you see? There we go. Yeah. Yeah, I'm not in full function today. So, what does endogenous mean? Originating within, native. And of course, an analgesic, we've already indicated, is inhibiting the pain. And the body has a variety of, these are chiefly peptides, endorphins, enkephalins, dynorphins. They're released at various points in the brain. They generally bind these dopaminergic stimulating opiate receptors, but in so doing, tend to diminish the sensation of pain. Because remember, we talked a little bit about this before. I'm going to put you in charge of pain minimization today. What are the possible ways you could do this? How can you reduce, in an anatomical fashion, not so much chemical, how can you reduce the patient's pain anatomically? All right, so Bob says prevent the signal from ascending from the nociceptors. That sometimes works, but again, because of the persistence of the pain, sometimes it doesn't. Remember, where is the pain? You need nociceptors to experience pain, but the pain itself is actually in the brain. I wanted to say Spain because I was thinking of My Fair Lady, but yeah, it's in the brain. So you could also block it in the brain, but sometimes one or the other, sometimes both are required. Sometimes the deep brain stimulation is helpful. There are neurological stimulators for the back. There are some for the brain. All right, let's stop there.